Um, okay, well, thank you, first of all, everyone, for coming and joining me in whatever time zone that you are in around the world. And thank you, Hugo, for organizing this amazing event. This is really fantastic. You know, obviously, everybody is in a very unusual situation right now, and this has really been really nice for feeling a bit less isolated and feeling really inspired by great science. And so it's been really lovely to see the talks of the last few days, and I'm really excited to talk to all of you about some of the work that we've been doing in my group over the last many years. To start off with, I guess I'll start off with sort of the same sentiment that Dean and Benedict started off their keynotes with, which is that one of the most interesting things to do in the history, or in biology really, is to think about biodiversity. We're surrounded by it, and it obviously is very engaging, but understanding diversity is extremely important because it obviously is important for a lot of vital human functions in human society, but also because there's going to be huge changes going forward. And so as much as we can contribute to the understanding of the processes that shape diversity, we can help predict and hopefully mitigate those effects going forward. And so for people like me, and I'm sure many of you, who think about diversity on kind of these broad scales, one of the first things that becomes very obvious is that diversity is not evenly distributed across space, time, or the tree of life. So when we look at the world today, like around here, this is a species diversity map. When we look at this map, we can see there's a lot more species in the tropics and in the pole. When we look through deep time, we can see that there's periods in the history of life where we have much higher standing diversity and other periods where we have lower diversity. We can see there's rapid changes to that diversity through time. And when we look across the tree of life, of course, we can see huge differences in the amount of diversity in different clades. So when we think about these different patterns and why we have these different patterns, there's a lot of interesting questions we can ask. In fact, that has been the guiding question of my research program over the last 20 some odd years, is trying to understand these processes that really shape organismal diversity across large scales. Now, there's a few things that I always try to keep in mind when I'm thinking about biodiversity and the evolution of diversity. And the first point is this one, and I think this is an incredibly important point that I know many of you also think about and work in. And that's, of course, that the vast majority of organismal variation is extinct. And these extinct forms aren't simply kind of intermediates of living groups. They're actually a far greater variety of forms than we could possibly envision if we just look at the modern diversity. And so as much as possible, we want to try to incorporate this variation, this extinct variation, into our models or into our analyses of the evolution of diversity. Now, it's not always possible. In many cases, they're not going to be well preserved enough in order to incorporate into studies like most of the ones that I'll be talking about a little later in this presentation. But we want to try and keep that diversity in mind, even if we can't directly sample it. Because if the mechanisms or the processes that we're invoking to explain diversity through time or diversity in the modern world can't also explain that variation, then they are probably wrong. So we want to keep that in mind as much as possible and incorporate those data when we can. And indeed, several studies have shown that incorporating even a relatively small amount of fossil data into analyses has a really strong impact and improves accuracy in terms of macroevolutionary analyses. Now, of course, time is not only deep, time is also developmental. And so it's also really important to think about developmental processes. And of course, we heard a lot about this yesterday. But it's really important to think about these developmental processes when we're thinking about the evolution of morphology. This was put really nicely by Conrad Waddington when he said the evolution of organisms must really be regarded as the evolution of developmental systems. And of course, here's a stereotypical example of an axolotl looking quite different from its closest relative. But in fact, the developmental change that's happening to produce that difference in morphology is actually relatively minor. So we want to try and consider that or keep that in our models or incorporate the information as much as possible, because the information from development may radically transform our understanding of the evolution of morphology. So we can do this potentially by, as Benedict and others were discussing yesterday, sampling through morphology and quantifying morphological change through ontogeny, and of course far more complex things as well. But going back to the previous point, it's also important to remember that this sort of information isn't usually available for wholly extinct taxa. There are rare cases where you might get some embryological material, but there are a few species here and there across the history of life. In fact, getting this sort of information, even for most living taxa, is incredibly difficult. Most rare taxa will not have any ontogenic data available. And in fact, most taxa will have relatively limited ontogenic data available. And so in trying to reconcile these different factors that are really, really important for understanding the evolution of morphology is what really led me into the field of morphological integration. And we've heard a lot about morphological integration over the last few days, so I'll just briefly recap. But it's basically, as developed by Olson and Miller, based on this really intuitive idea that evolutionary changes in one trait do not occur independently of changes in other traits. And by quantifying morphology across different taxa, we can, or quantifying morphology across different structures, we can actually identify the relationships among these traits. So for example, if we quantified the entire skeleton of, in this case, an elephant, we might find that all the different structures are very highly integrated and they evolve in a correlated fashion, they vary in a correlated fashion. What we usually find, though, when we actually quantify complex structures is that really traits tend to fall into groups that have relatively strong within group correlations or covariances, but relatively weaker associations across different structures or across different parts of a structure. And so we would term these modules, these semi-independent groups of traits that evolve in a fairly tightly integrated fashion, but relatively independent from other groups. Now, this field of integration and modularity has a really rich history of study. We heard some really nice talks going through that history over the last few days. And of course, there's been a lot of attention paid to what really drives these patterns of trait interactions or trait integration that we find across the diversity of organisms that people have looked at. And trying to tie those back to associations of genetic traits, of developmental processes, of various shared associations or associations that arise at different points through development. And that is a hugely interesting field of study. What's most interesting about it to me, as somebody who's a paleontologist coming from the perspective of only ever having adult morphology, it's the idea that you can access genetic and developmental information when all you have is the adult morphology of, in many cases, an entirely extinct organism. So that feels like a very powerful tool, potentially, to try to understand the different processes or the different factors that have shaped the evolution of form. The other thing that's really interesting, of course, is that these relationships are a primary influence on how morphologies evolve and how they vary. And so looking at any individual character in isolation will give you a very different picture than if you take into account these relationships the different traits have with each other. And how these relationships among traits evolve is in itself a really fascinating topic with a lot of interesting hypotheses that have been posed. And if we look at this figure here, you can see that there's essentially a few different ways, very simplistic ways that one can think about this. But one idea that people have repeatedly suggested is that more integrated phenotypes are the ancestral condition. And through time, what you see is a fragmentation of these phenotypes into more modular organizations. And these modules can then vary independently and evolve independently, potentially exploring a broader range of morphospace because they can then respond to maybe different selection pressures. On the other hand, you may find that over time some traits become integrated, potentially as a response to functional associations of those traits. And so understanding how these relationships, how they stand in different clades, but also how they evolve is really important if we want to try to understand the evolution of morphology and why morphological diversity is structured the way that it is across the modern world and across the past and through the past. So just to kind of briefly summarize that kind of in a very simplistic way, how these trait integrations or trait relationships can influence morphology. A mathematically obvious thing is that they limit variation to a relatively restricted area of morphospace, aligned to the principal components, of course, which are defined by the covariances. So if you just take this simple plot here, which has been reconstructed or drawn in a million different ways, but here's my take on it. In gray circles are phenotypes that have no integration among the different traits, and they can just disperse through morphospace in all these different directions. And in yellow and blue, what we have are traits that are integrated, and the expectation of morphology is for integrated traits. And of course, they align to principal axes. But what's important to point out here is that two things. One is that, of course, a huge amount of morphospace is left unexplored due to the integration of traits. But you also see that traits may actually, this integration among traits actually may push variation farther along those principal axes than we might expect if those traits were unintegrated. And so you might actually find more disparity along certain axes than you would if there was no integration of those traits. So integration may result in sort of both ends of this spectrum. There's a couple of things I think that are also important to point out when thinking about this plot. Again, it's a completely obvious plot, but it, I think, makes a couple of points very obvious, which is that things like, because of variance being pushed into a few preferred directions of shape change, this will likely have the effect of increasing homoplasy and convergence among taxa. And I think this is an important point because convergence is obviously a very interesting phenomenon. It's something that people have studied at great length. And is often treated as something really unexpected or something really special. And I think it is special, but I don't think it's unexpected. I think if you take into account the relationships among traits, if you take into account shared developmental pathways of taxa, then actually convergence under a similar set of selection pressures is probably the expectation rather than the exception. So I think that's something that's interesting to think about in terms of our perspective and how viewing morphological evolution through this lens actually might change our perspective on what the null of different expectations is. And one other point is that integration of traits may not influence disparity in rates similarly. They may, but they don't necessarily have to. And this is something that we explored a bit in a paper a few years ago, and we called it the fly in the tube model. And essentially, you can imagine this tube that this fly is stuck in as the area of morphospace that is being defined by your trait interactions, your trait integration, the covariances. And so the fly is kind of stuck within, the fly is, morphological evolution, and it's kind of stuck within this relatively narrow range of possible shapes. But within that range, it can actually zip around at different speeds. It could evolve quickly, it could evolve slowly, but it's kind of stuck in this area. And one empirical example of this might be something like the carnivoran cranial base. And this is a region of the skull that's often treated as very conservative, very good for phylogenetics. But actually, if you quantify it, yes, it doesn't show very much disparity compared to other regions, but actually its rates of evolution are not low. And what that suggests is that even though it's stuck in this relatively narrow tube of possible shape space, it's actually evolving quite quickly within that. It's just re-evolving the same forms over and over and over and over. So rate and disparity may not necessarily have the same relationship to integration. Okay, so there's a lot of different options that I've thrown out here. There's different ways that rate and disparity might be related. There's different ways that disparity might be impacted by the integration of traits. You could expect more, you could expect less. So really what we need to figure out is which of these effects actually dominates in the real world. And in order to do that, we need to gather empirical data. So what we've been doing in my lab over many years now is trying to answer a bunch of questions, including, among others, how do trait interactions really influence the tempo and mode of phenotypic evolution? Do they reflect ecology or life history? So do we see similar patterns of trait integration among things that metamorphose or things that eat plants? Are some of these modes potentially more evolvable, or more modular? And is there this, this kind of really interesting question. Is there an evolutionary trend for increased modularity through time to circumvent some of the constraints of trait integration? Now, I don't know that I can actually answer any of those questions, but hopefully, in another 20 years, I'll be able to. And all of this is part of a, much broader set of interests in my lab, and I think more generally, to try and understand what are both the intrinsic and extrinsic factors that influence the tempo and mode of evolution for organismal form. That is a really fundamental question that's important not just for people like us who are interested in why diversity looks the way it does, but also going forward to try and understand how diversity will respond to major biodiversity and climate crises. So in order to do that, what we have to do is really try to quantify morphological evolution and trait integration across taxa. And this is something that has been done. Of course, a lot of people here have talked about this. And this has been done, I would say, more than any other clade. This has been done within mammals. And many people, myself included, have spent a lot of time quantifying patterns of phenotypic integration within different mammalian species and comparing across the clade to see how conserved those patterns are. So primates, probably more than anything, have been studied by a lot of different people with a lot of different approaches that have really come up with really similar results, which is quite nice. And in fact, when you look across placental mammals, multiple studies now have demonstrated there is a fairly large degree of conservation in terms of patterns of modularity across the clade. There's some differences in magnitude of integration, but the overall pattern is relatively similar. There's a few exceptions, especially in the really weird aquatic mammal groups, but by and large, mammals are quite conserved. So when it comes back to answering some of those questions that I posed in terms of how modularity and integration relate to things like diet or life history and things like that, well, mammals may not be the best group. For certain questions, certainly they are, and there's they remain my absolutely favorite group, but they have too much similarity in many different attributes to be able to really get at some of those other questions. And so in order to answer these questions, we really must quantify and compare morphological evolution across a broader range of taxa. And so for the last few years, that's what we've been doing in my group. We have been scanning specimens across the world with CT scanners and laser scanners, traveling around to at least 50 some odd different collections around the world to put together a data set that right now numbers probably something around the order of 2,600 specimens, about 18 to 1,900 different species, and about a quarter of those are fossils. And on this phylogenetic tree over here, you can see the distribution roughly. So we have mammals over here, where we have both really nice sampling of extant mammal diversity, but also an incredible sample of their fossil history. We have over here squamates, I think that's crocodiles over here. No, turtles, sorry. Over here we've got all the archosaurs, including crocodiles, dinosaurs, birds, and over here the various amphibian groups. And we have lots of fossils towards the stem of this tree. Of course, some of the groups are much better sampled for fossils than others. But as much as possible, we have tried to incorporate the vast majority of well-preserved 3D specimens for these different groups. And in doing that, what we've come up with is, again, this really nice representation, I think, for the full diversity of form across, or near full diversity of form in tetrapods, so in all the limbed vertebrates. And while we focused really on capturing across the entire clade, for each of these different kind of class level clades, we have also gathered lots of within species data sets. So we can look at both phenotypic integration and evolutionary integration and modularity, and then compare across those levels. So that's the discrepancy between the species and the specimen level data sets, is that we have a number of species for which we have 40 or more specimens represented as well. And so that seemed like a really difficult thing to do, and it was in fact very difficult. It required a lot of people, spending a lot of hours traveling and scanning and cleaning up scans, and obviously a fair amount of funding. And then of course we realized that that was the easiest part of this entire project, because then once we had all that data, we had to figure out how to accurately quantify and compare that morphology. And as I'm sure all of you will appreciate, this is not an easy task. In fact, I would describe it as similar to falling into Sarlacc's pit. You know, a new definition of pain and suffering as you're being slowly digested. In our case, it was a couple of years of really trying to get our heads around how to solve this problem. But of course, the problem here is that there are limited comparable landmarks across clades. And of course, as you go higher and higher up in your comparisons, those landmarks reduce in numbers. So whereas you might be able to identify, say, 60-odd landmarks in the mammal skull, by the time, and in fact, in all of these different clades, most of them, we can probably identify, you know, 40 to 60 different landmarks that we feel that we can reliably identify in all of our different groups. By the time you get down to the entire level of tetrapods, that reduces by a huge amount. And in fact, the first exercise that we did in this project was to take these different skulls. Everybody in my group had a different clade they were focusing on. We took all of our list of landmarks that we had come up with for our specific clades and compared them. And the number that we came up with was 12, about 12 landmarks that we could identify across all these different taxa. So if you've just spent a couple of years putting together this data set of 2,600 3D scans, roughly, I don't know, £500,000 pounds in funding to get that data set together, 12 is not the most impressive number to go back to the EU with, although it is certainly the best thing that they have heard out of the UK in several years. Nonetheless, though, it's a very unsatisfying amount of data when you look at the variety of forms that we're trying to compare. And in fact, just looking at these images that are on the screen in front of you, you can imagine that those 12 landmarks exclude almost all the variation in that form. It gives you a very, very poor representation of those phenotypes. And the goal, of course, is to have an accurate, comparable, and high-resolution description of phenotype. We want a method that can actually capture that variation, not a method that has to exclude all of that variation in order to be comparable. And so, how do we solve this problem? And that's something that we tangled with for quite a long time, because we wanted to make sure that we are able to capture the variation in our data set in order to understand how that variation evolved. And a method that was going to exclude the most variable parts of the skull or the most variable clades was going to give us a solution that we are not going to be happy with. And so the approach that we've taken is this, and that is to take our homologous landmarks, our type 1, type 2 landmarks, which are in red up here in A on this bird skull, so these red points here, which we can identify across our different taxa. Then we use these what's in yellow, these are curve semi-landmarks that join up those different type 1, type 2 landmarks. And then what we do is we use a semi-automated process, so not fully automated like what Benedict was talking about yesterday, but something in between, because of course we're dealing with a really big variety of shapes here, and it becomes really hard to automate, to come up with something that works as an automated process, although we have tried. And what we do then is we take a template. Actually, what works best is a very generic template we found. And we take that template and we apply surface semi-landmarks onto that template and then morph that template essentially to all of our specimens. And so here's the specimen being warped the template being warped to the specimens using those common landmark and curved semi-landmark points as anchors, and then inflating them off of the template and onto the specimens. And that gives us a really nice representation of the morphology of those specimens. And what we end up with is something like this. So this is just a sample of some of the skulls in our data sets. Now in this case, these are slightly different templates for each of our data sets, because in our first pass, what we're doing is having, for our clade-specific analyses, we have slightly different templates to maximize the representation for those individual clades. In some cases, those are not going to be comparable at the higher levels. But you can see that broadly, we have a similar representation of the morphology across these different clades. And it's a really nice, dense representation of those shapes that really allows us to capture not just suture points, not just the tips of processes, but some of that ornamentation that might happen, differences in curvature, just a huge amount of variation in morphology that can't be captured by just the landmarks alone. And what I also like about this is that it means that we can also separate our data set into the different regions, which means we can start to think about things like integration and modularity, but we can also look at patterns specific to different parts of the skull. Because this is usually the point in a talk where somebody will ask me why we didn't just use a fully automated approach. And this is the reason that we don't do that, is because we want to be able to identify where in our different specimens our landmarks and our semi-landmarks are located. We want to be able to look at the relationships among those different regions, and we want to be able to partition the skull into those different regions. So what we end up with is this really, I think, nice representation of the skull that can be separated out to the different regions, and then we can do lots of interesting things with that. But of course, we got this far and we thought that would be the hardest part after we thought the previous part was going to be the hardest part, and then we realized, actually, nope, it's going to get harder again. And that is because, of course, we need to analyze these data. And as Dean talked about a few days ago, dealing with big data or high dimensional data is difficult. The methods that have been largely put out there are not meant for these sorts of data. They actually largely can't even handle these sorts of data. So most of the methods that are being developed for analyzing microrelationship patterns are really focused on univariate data. In the cases where sometimes they'll say that they are multivariate methods, but usually they've been tested on something like three traits. We're obviously looking at something far greater than that, and so this becomes really problematic. And so we've spent a lot of time trialing different approaches, trying to figure out what works, trying to figure out what the differences are between the results we might get versus results if we use our landmark only data. So we usually do our analyses both ways and compare the results. And also trying to assess what we gain by doing this extremely time-consuming and difficult approach. Obviously, if there are many times where I think we should have just stuck with a 12. We would have been done with this project years ago. But what we have done is taken a look at how much more information do we gain, or rather, how much information do we lose as we subsample down to landmark only data sets or landmark plus curve data sets. What we find is that indeed we gain a lot of information using these additional surface points. Now in this plot over here, this is essentially sort of a collection curve where you're looking at the fit of the analyses if you as you remove landmarks. And so as you go down to fewer landmark numbers, there's a big decrease in the fit, but usually starts to plateau at about 20 some odd landmarks. So we could see that we were actually oversampling by about double. And another point here, at least for these data sets, so this is tested on the Sicilians and on the squamate data sets. These are clades that don't have tons of ornamentation on their skull, so they don't have lots of horns and things like that. So they are probably slightly underrepresenting the amount of information that you gain with the surface data. But in fact, by just using the landmarks and the curves, we do actually get a huge amount of the signal. The vast majority of the signal is in the landmarks plus the curves. And for these clades, the surface points actually add a relatively minimal amount of additional information. Now, we don't get that same result when we look at things like mammals and other clades that have lots of crazy things going on in their skull. But I think this shows that these big data are worth it, that we are gaining a lot more information about morphology by having these more heavily sampled in terms of landmark and semi-landmark number data sets. And so, like as I said, we've done a lot of work to try and figure out what we lose, what we gain with these different data sets. But what we gain is the ability to then look at some really interesting questions with a really accurate and detailed representation of the morphology, where we're not losing a lot of the variation because it's too variable, right? If we want to understand variation, we have to capture the real variation in our data sets. We can't just artificially dump it out because it's difficult. And so once we have that, we can start doing these really cool analyses. And so I'm just going to walk you very quickly through some of the work that we've been doing in different clades. So for example, this is our analysis of birds that we published in the last few years. And one of the things that's really interesting about birds that's come out in a number of papers over recent years. So I think the first time I was aware of this was a paper by Jen Bright and colleagues that was super interesting and suggested that diet and shape of the beak was not strongly, or shape of the skull, I think, were not strongly correlated in birds. And this really, I think, surprised a lot of people. It certainly surprised me, because of course, a lot of the early ideas about evolution were in fact inspired by beak shape and diet in birds, Darwin's finches. But in fact, so we ran this analysis with our full data set, and we found the same result. In fact, diet and shape are not strongly correlated in birds. But interestingly, what we did find, and I think this is similar to something that Dean mentioned a few days ago, is that rate and diet are strongly correlated in birds, with specific dietary groups showing faster rates of evolution. In this case, it was the seed eaters and the nectar eaters that show faster rates of evolution in a lot of the parts of the skull. We also found that different parts of the skull show really different evolutionary rates, and we suggested that this could potentially be related to the different embryonic tissues that give rise to those regions. So things that have a lot of input of the anterior mandibular cranial neural crest show higher rates of evolution. Also things that tend to have more contribution of different tissue types appear to have faster rates of evolution and lower within module integration. So this is a hypothesis about developmental causes that can then be taken forward and tested to really find out if that's true or accurate. And we can also look for that in other clades as well. One of the other cool things that we can do with this data set, again, is compare things that are really difficult to compare with landmark only data sets because of how different the actual type 1 landmarks might be, but the structures, of course, are still similar or homologous. And so what we can do is take things like that bird data set and expand it out to the full bird clade, or the stem birds, the dinosaurs, and start to compare how different regions of the skull are evolving in birds versus dinosaurs. And what we were able to demonstrate by doing this analysis, and this is over in these figures down here, these are the dinosaurs and these are the birds. And what we've been able to demonstrate is that across really almost all the different parts of the skull, birds are evolving in terms of shape at a much slower rate compared to dinosaurs. Now, if you know a lot about dinosaurs, that might actually feel about right, because dinosaurs are actually incredibly diverse in terms of morphology when you can think about skull morphology. And a lot of that really probably relates to the fact that they have a much wider variety of feeding mechanisms. They use their skull, their bony elements of their skull, for display in a way that birds only use soft tissue structures like feathers. And so you have a lot more going on in dinosaur skull evolution. But here we're able to quantify that indeed birds are evolving in terms of the bony morphology of their skull in a much slower rate compared to dinosaurs. And I think solidifies the point that birds are terrible dinosaurs. I'm a mammal person though. We can also look at this in our squamate data sets and again, compare things that are really difficult to compare normally. So we're looking at snakes and lizards and various extinct squamate groups like mosasaurs, and able to combine them in a single analysis where we can identify things like a high degree of convergence among fossorial forms. So that's this gray area of the morphospace over here. And we can also identify interesting things like the fact that herbivory seems to be consistently associated with high rates of evolution in lizards across most of the skull. And interestingly, when we look at that diet and shape relationship that we were talking about in birds, if we look at that in this group, what we found was that whereas diet and shape are strongly correlated in the anterior rostrum, so things like the premaxilla and the maxilla, in lizards, when you look at snakes, those regions don't show a high correspondence to diet. Rather, the shapes of the bones associated with opening the mouth really show a strong dietary signal. So different parts of the skull show different relationships to ecology. And again, having this sort of data set really allows us to capture that. We can talk about frogs. One of the things I've realized during the course of this project is that amphibians are in fact the most interesting group of vertebrates by far. And some of our work in frogs has really surprised us in that we were expecting to see a really strong impact of things like life cycle on frog morphology. But in fact, what we found was that life cycle doesn't really discriminate different frogs very much in terms of being having active feeding larval stages or not having that kind of independent feeding larval stage. But what in fact really has a strong influence is habitat. So aquatic and fossorial frogs really evolve at much faster rates than other ecological groups. And we interestingly also were able to show that the timing of when bones develop in frogs also shows a strong correlation with how fast they evolve. So late ossifying bones actually seem to evolve at a much faster rate compared to some of the early ossifying bones. Now we can also look at this in salamanders. And salamanders are, of course, an incredibly diverse group. They do lots of really interesting things in terms of life history, in terms of habitat. They have this incredible range of cranial morphologies that are shown here in this circle diagram in the middle. And again, a lot of the most highly variable parts of the skull are ones that you would not be able to capture with landmark-only approaches. But we're able to do that. And what we were able to show, in fact, using these approaches is that the exact opposite of what we see in frogs seems to be the case in salamanders. Life cycle is a really strong discriminator of shape of the skull in salamanders, where you have pedomorphic forms in this area of morphospace, and this is really a gradient to degree of pedomorphosis. We have direct developing forms over here, and we have biphasic forms over here. This is the ancestral life cycle for this clade. But in fact, you see, at least in this data set, where this is a sampled at the genus level for salamanders, at least 11 transitions in life cycle type. And what we found when we reconstructed rates of evolution for this data set over here is that shifts in life cycle are consistently associated with shifts in rate of cranial evolution. And in fact, we also were able to partition this out by the different bones again, but also by different life cycle, and show that direct developers consistently evolve at a slower rate compared to the biphasic and pedomorphic salamanders. So we see this really, really interesting difference between these two groups of amphibians, or the stronger impact, the stronger influence of life cycle is what we expected to see in frogs, but in fact we see that in salamanders. Habitat, on the other hand, doesn't really show much of a pattern in salamanders, but of course with a stronger influence in frogs. And so we have this really unexpected result in that data set. And so, overall we have now analyzed about 1,400 species across this data set, and been able to really start to look at commonalities and differences in terms of the different factors driving shape evolution and diversification of shape in these different groups. Of course, we can also compare across them in terms of pattern of integration, and this is something that we're not doing directly because, of course, of differences in the data sets. And what we were trying to do is see whether there were any commonalities in the overall pattern of modularity. So the different elements of the skull, do they show similar associations or quite a lot amount of differences across different groups? Remember, within mammals, this is fairly well conserved. But in fact, what we found is that there are some similarities across all of these groups. So these network plots with the strength of the line showing the degree of integration across the different elements. In most of these groups, actually pretty much all of them, we find that the occipital regions and the suspensorium show strong patterns of stronger integration. But in terms of the facial region of the skull, that tends to be a little more different across the different clades. So there's a lot more variation in terms of facial integration, whereas essentially the basic cranium and the suspensorium show a lot more similarity across these different groups. Now that's something that we're obviously going to continue to explore and more direct comparisons, but it's an interesting first result and not exactly what I expected, which is often the case. So to kind of wrap up here, I hope that I've convinced some of you that really these high density approaches are really a powerful tool for comparing morphological evolution across disparate groups, and really for assessing the influence of both intrinsic and extrinsic influences on shape and morphological diversity, really allowing us to really capture the real variation in our data sets without having to just exclude the parts that are indeed the most variable and in many ways the most interesting parts of our data. Trait integration can significantly influence morphological evolution by constraining variation to limited areas of morphospace, but also potentially by pushing variation into a more extreme areas of the lines of least resistance. So quantifying trait relationships is very important. And also thinking about how those would impact a lot of the phenomena that we tend to think about in morphological evolution, I think is really critical to accurately reconstructing the evolution of diversity. Both development and functional interactions are really major influence on integration, and also, of course, in morphological evolution. There's a lot of variation across tetrabiotic clades in terms of whether both the patterns themselves, but also in terms of whether diet or life history really are dominating in different groups. Vertebrate skulls are highly modular, but not really all in the same way. There are some consistencies, though, especially in regions like the occipital region and the suspensorium, where the face seems to show a lot more variation across the different groups. Now, this is, of course, not the entire data set analyzed. We still have a lot of the mammal groups analyzed, since I'm in charge of that, so it takes longer, of course, as well as turtles and a few other things. So stay tuned for those over the next year. And looking forward, we have started expanding to new systems. So like every project, this one has really inspired more new things than more new questions than answering any of them. But we've started a new project looking at pelagic fishes and arthropods, 2 new projects rather. We're also, again, this is a very time-consuming method. And it also, raises a lot of challenges, both in terms of data collection and in terms of analyses. So we are working with several others in terms of trying to improve both our approaches to data analysis, but also trying to automate the data capture as much as possible while still being able to answer the questions that we want to answer. And some of the things that we've been thinking about or playing around with are actually entirely surface-based approaches, such as the Sinatra method that was described earlier this year. And what we really want to go towards, now that we have some idea of how variation is distributed across these different groups, we have some idea of the commonalities, what we're trying to do is work with developmental biologists to start to try to interrogate the mechanisms underlying those patterns, because now we can more precisely pinpoint the areas that are especially interesting, and specifically which groups those areas are showing a great deal of variation or a great deal of or faster evolutionary rates. So those are the directions that we're moving towards. And so with that, I'd really like to thank my really amazing lab who figures out how to do all of this work. It's a real team effort. It takes everybody kind of scratching their heads and occasionally banging them against walls to try and get all of these methods and all these data together. And they've been real troopers. So thank you to all of them. We have a huge amount of collaborators that give us access to some amazing data sets, but also help us with through methods and trying new things and refining our ecological and life history data. Thank you, of course, to all of the funding agencies that fund this work, especially the EU, and especially to all the curators and collections managers around the world who give us access to these collections. This work is not possible without the support of those institutions, and without those institutions having the ability to maintain those collections and to make them accessible to researchers. And so it's really a great service that they're doing for the entire world. As we analyze all these data, we're putting all of the scans online. So I think we have about maybe 900 or so scans from this project available already on Phenom 10K. So all the birds, quamate, and amphibians are already up there and freely available to download. So by all means, go and use those data for your own projects. And I just want to end with a quick parting thought, if I may. Sorry, I realize I've gone a little bit over here. But this, the quote at the bottom here is one that I really like from a paper by Mike Collier and others about parametric degrees of freedom do not constrain natural selection, so why should describing the phenotypic response be so constrained? And this is probably not how they initially intended it, but to me, this quote really is a call for innovation in the field and to think beyond the kind of the boundaries of the morphospace that are defined by the methods that have come before us, so the people that have worked in this field before us, and to really try new things, to really innovate and try and broaden the field to address really important and interesting questions in biology. And as Benedict said the other day, morphometrics is sort of a perspective, and it's really important to bring that perspective to other fields of biology because it is a very powerful approach and powerful way of thinking about variation and morphology and evolution. And as part of doing that, as part of really supporting that innovation, I think the field needs to really be open-minded and more inclusive. I think this has come up in the last year in Morphomet and other places, but of course more broadly, it is well established that science is really suffers from a series of biases. And in the last few weeks, as we all live through this really quite extraordinary event in human history, it's been brought up that these biases are actually being amplified by the current situation. So as we all, are sitting in our houses, not able to go outside, either working on papers or chasing your kids around or doing whatever, I hope that we can all take a moment to think about how we as scientists, as collaborators, as reviewers, as editors, as colleagues, can really promote the innovation in this field, promote inclusiveness in this field, and really try to make this as professional and supportive and innovative a society or a group of workers as possible. And I think if we do that, we can take this fairly negative global situation and really make it a positive transformation for science by trying to really promote a better professionalism. And really, I think if we do that, we'll be able to make sure that morphometrics, geometric morphometrics, is able to innovate and really rise to the challenge of addressing these very fundamental ideas in evolutionary biology and in biology and the study of the natural world. So I will leave you there. Thank you very much.
Thank you very much, Anjali, for your fantastic talk. I really enjoyed every second of your talk. And I received many messages, so probably then I will share with you that they're all fantastic. Is all the slides you are showing. Is now opening for questions. You can raise your hand. I know it's allowed. Let me check, Anjali, if allow the raise hand function.
Do I need to do something? Should I stop sharing?
Pueden preguntar. You can listen. You hear me, Anjali?
Yeah.
Okay. So, para las personas que desean preguntar en español y quieren que traduzca, pueden hacer la pregunta en español y yo puedo traducir a Anjali.
Oh, sorry. Sorry, I didn't quite understand. Sorry, no, I actually couldn't understand.
There's one question now. I will unmute. Okay, Boris, you have.
Hi, can you hear me?
Yeah, I can hear you. Thanks.
Anjali, I am really emotional for your presentation. Was really good. I really like it.
Thank you.
I would like to ask you if you do reconstruction or do your analysis with the material fossils or not.
With material fossils?
Yes, with material fossils.
Yeah, so we go out and we scan the fossils in 3D using our surface scanners usually, or CT scanner where possible. So we've gone to all these different collections and scanned the fossils, and then we do the analyses on the 3D reconstructions of them. And as much as possible, we put them online so that other people can also benefit from that, because obviously getting access to those fossils is not always very easy.
Okay.
Oh, sorry, I think, were you actually asking whether they were reconstructed or in terms of any deformation?
Mm-hmm.
Oh, yes. Okay, so in that case, we either use fossils that are completely undeformed, which is, of course, a big constraint on our fossils, or we work on fossils that have been retrodeformed or reconstructed by some of the people working in functional mechanics. So these are people who have usually done a huge amount of work trying to reconstruct the fossils very carefully in 3D and done a lot of sensitivity analyses on some of the different options. So we only use ones that are kind of either perfect or have been retrodeformed under very strict criteria. You can also drop out, if they have some missing elements, we will sometimes just analyze the elements that are available and drop out the ones that are missing.
Okay, so I will move to the other question now. Erwin, you have the word. Can you talk now?
Okay, can you hear me?
Yep.
Yeah, okay. And first place, thank you for your impressive presentation. Well, I work with crustacean, the Cabot crustacean. In this group, I have two great taxa, groups of species. The Anomura, yeah, with 5,000 species, with the most diverse types of body plans, and are cephalothorax, highly modular cephalothorax.
Mm-hmm.
On the other hand, the Brachyura group have 15,000 species with the body plan very similar bit with them, and their cephalothorax is highly integrated.
Yeah.
Based in this evidence, it's possible to consider that the highly modular structure has a promoter of variability, phenotypic variability. Meanwhile, the highly integrated structure can be considered as a promoter of highly diversification of species. On other hand, the modular promotes the phenotypic diversification, and integration promotes the diversification of species. What do you think about this?
I think that's a really, really interesting idea, and that could very well be the case in some clades potentially. I think we just don't know enough empirically to be able to know whether either of these different effects actually dominates, or if you can see the full range of effects. But I do think that specific one in regard to arthropods is a really interesting one. Because of course so when I first moved to the Natural History Museum, part of the reason is because I wanted to start thinking about insect body plans and diversification as it relates to modularity and integration. And I remember at my first Christmas party there, Greg Edgecombe and I were talking, and I said, well, I want to look at insects. And he said, insects are really boring. They're so constrained in terms of their morphology compared to all the other arthropod groups. So the question isn't, why are insects so diverse? The question is, why are they so much less diverse than their close relatives, even though they have so much more taxonomic diversity, which I feel like is sort of getting at the same thing as what you're saying. Of course, insects are hugely diverse in body plan also, but certainly some of their close relatives show much greater differences in terms of structure. So I think that's a really interesting idea. I haven't thought about it in exactly that way before, but it would be really cool to follow up on. And if you want to talk more about that or potentially collaborate on things looking at that question, let me know, because that would be really interesting.
Fantastic. Eric, you can talk now. Now, Eric, can you listen? Can you talk? Eric Mesquita. Well, I think I cannot hear. Anjali, I will ask you a question, probably if you can recommend all the people that are watching you now. Mostly of all wanted to do 3D morphometrics, but they don't have the opportunity to have the technology to scan all the organisms. So they mostly ask for collections to having the scan, all that. I saw the website for Phenomic website. So how do you can give some advice to people that wanted to start working in 3D morphometrics? And what is the best way to start on that, and if they can practice downloading, for example, the scans and what you have to do.
Okay, sure. Yeah, so that was very much the issue that you raised is very much the reason why I created this database, I guess about 7 years ago or so now. Because, you know, I have, in many cases, have had the luxury to be able to travel around and gather all of these scans with 3D equipment, but that's, I think, a real rarity. And of course, I do what I want to do with these data, but there's a million other projects that people could do, or people could obviously replicate what we're doing. And so putting those out there so that other people have access to this, I think is, you know, a real driving force of a lot of our efforts. And so, unfortunately, I'm not alone in this. Over the last few years, there's been more and more attention paid to making these data available so that people are not limited by what their ability to travel, which of course is more and more important today, and by access to specimens. And so, you know, for Phenom 10K, you can just go on. All you have to do is, like, you know, make an account with your email and password, and you can just download anything you want completely free. You don't have to ask permission for anything. So you can download, those are either STL or PLY files. You know, there's a mix of them, and they're relatively small size so that people also aren't constrained by having, you know, different internet connections and things like that. So you can download those sorts of data. There's also places like, there's a number of websites now that are doing this. So MorphoSource, which is based in the US, is of course a really huge effort. And really, if I had started this project 5 years later, it would be a different project because we wouldn't have had to have spent all that time collecting at least the extant data, and we could have just focused on the fossils, because they will have, you know, basically every genus of vertebrate scanned within a couple of years. And that means that that will no longer be a constraint for people and for what they want to do. Well, you can, yeah. But so with MorphoSource, a lot of the things that are being put on are just the raw image stacks and not the reconstruction. Some of them are reconstructed. And so then you have the problem of being able to reconstruct that. But there are new packages or existing free software, because of course CT reconstruction software can be very expensive because it's medical. But you can use things like 3D Slicer, is an effort that both Dean and I are involved with in terms of building in some morphometric tools into this free software called 3D Slicer. And there's also courses that they're running to help people learn how to use it, and also to help people learn how to do morphometrics using that interface, but also more broadly. So I think those are really good places to start to get your head around 3D data. But a lot of it is just, you know, downloading some data, getting a feel for it, and using some of the free software that's out there to start playing with landmarking. So 3D Slicer is a good tool, and obviously there's tools in R also that can help with landmarking and things like that.
Fantastic. I have one last question, Javier.
Hi, can you hear me?
Yep.
Okay. Thank you for your amazing talk. And I'm curious, how do you deal with missing data, particularly when incorporating fossil specimens? Could you include a fragmentary specimen, and how complete should it be to be incorporated in the analysis?
Yeah, okay, so there's a couple of different ways you can deal with missing data. And it really depends on how much is missing, and really whether that fossil, for example, is something that has a close relative, or a phenotypically close and evolutionarily close specimen that is complete in your data set or not. If it's, so what we do is we can drop out those missing data from our analyses in the individual region. So because we're doing like individual region analysis, we might be doing just the maxilla or just the frontal or, you know, just the occipital. And so we can include all the taxa that have that bone in that data set and drop it out from the other. So that's one way of doing it. It's not entirely satisfactory, but it's one way of doing it. We can also estimate the missing data using tools and things like Geomorph or Morpho. And you can do that making a couple of different choices, whether you want to use it off of a shape or something that's closely related. There's a couple of different ways of doing that. So you can estimate your missing data, or you can exclude that specimen from the analyses of a specific region. So we've done it both ways, depending on whether we're basically based on how big of a stretch it is to estimate that missing region. If we think it's something that's basically going to be the same as this thing that's right next to it for all the other regions, or it's a very close relative, then we would estimate. I would feel okay with estimating it. If it's something that's quite unusual, then we'll just drop it out of the analyses. And unfortunately, we've had to drop out, so there's about 400 some odd scans that we've gathered that are not making it into our final analyses because they are just too, there's too much missing data, and it doesn't make it really worth the time at present, because of course we have a massive amount of data. But over the next several years, what we're doing is, I'm getting master's students and PhD students to really focus on one, doing those really complex retrodeformations to try and incorporate those, or also to do more of the region-specific analyses that can take advantage of those. So depending on what your data set looks like, you could estimate them, or you could leave that specific region out. But if it's too much, then you may just have to unfortunately drop out the whole thing.
Okay, Anjali, I would like to thank you again for your fantastic talk. We have more than 470 connected people now watching you.